Good morning, sir. Yes, sir.
Okay, thank you. Yes, good morning. We will discuss about transgenic mice or transgenic animals. So one thing before is I still don't have my graphics tablet. So I'm going to talk about transgenic animals today. And I will also share some of the other videos that I have that I recorded. And you can also look into a lot of other resources as well. Why I'm taking this transgenic mice is in the last class we were discussing about piggyback transposon based nuclear reprogramming and how to make transgene free induced pluripotent stem cells. So these two can go hand in hand. So I thought I will discuss both. In this case, I would want to discuss a little more details about how it is done, right? So transgenic animals is, I want to discuss that now. So I will not use much of my pen and I would not be able to draw as much, but still we will get through a lot of details and that should be sufficient. First thing is that transgenic animals, when the majority of the time we do with mice, okay, that means it's as if it's a proof of confidence, proof of principle that can be done with many other organisms as well, with other mammals, including humans. But it is, of course, regulatorily prohibited or highly controlled. So the first thing we should be in a state to define what is transgenic mean. That means there is a genetic manipulation. Transgene usually would mean that you put in a gene from other gene outside source and put it into a genome. And these are, that means you have introduced a foreign genetic element in the cells, including the germline. That means if there is a mouse, the mouse should be having all the cells of the mouse should be having this transgene. Then you call it a transgene. That means the somatoplasm as well as the germ plasm should have had this gene. Only then it is inherited to the progeny. So towards constructing this, that means we are actually, as the name, the definition here implied, we are making a recombinant DNA. We are making a recombination. So first thing is the design of the gene construct. I'll upload a video which I was trying to explain on the board for somebody else, and I think it will be useful there as well. It's on the classroom, so it is a little bit different, of course. And then how do we put it into the cells? Then how do we raise the organism from it? From modification of the gene, constructing recombinant DNA, putting into the cell, raising an organism from it. And then how do we characterize? And then how do we study? The rest of the things is about study, how much is expressed and other things. But from here to here, we have a lot of things to perform. So the overview is something like this. There are two popular methods. One of it is called as the direct microinjection method. The other one is called as the engineered stem cell method. The first one is we take DNA and we take the DNA and directly give it into the fertilized egg. And the egg will give rise to a morula, then blastula, and then it will give rise to an organism. If the gene that we put in has got incorporated, then we would have a transgenic animal. So the first one is DNA microinjection. The second one is the engineered stem cell method that will go through this process. It is a complex process. We have to make the DNA, recombinant DNA in the vector. Then we should put it into the embryonic stem cells or embryonic stem cells. And then we need to put it into, the transfer and put it into the blastocyst. And then we would get a chimera, and from chimera, we will, from this will be a chimera, and we will discuss about what is a chimera, and then we will get the organism. Let's see in, okay. Yes, Yogeshwaran, thank you. One second. That is what I was asking. Did we discuss about this whole PPT? Can you please?
Microinjection method and engineered stem cell method also we have discussed, sir.
Okay, that's good. I actually asked somebody about it and they said maybe we didn't. And that's why I was taking. So we discussed about positive selection and negative selection.
About that, I'm not sure about that topic, sir. Could you please again tell us about that?
Yes. Did we discuss this? Yes, I will just play the PPT and you can tell me if we have. Did we do this? Anybody, please?
No, sir, we didn't discuss it.
Okay. I'll just still scroll through anyway, and probably you can see others also. I would want so that we can. Did we discuss about this one? Any idea?
No, sir.
No. This one or this one? No, right? Or
Yes, sir, we done it, sir.
we have done
This
it?
one we have done, yes, sir.
Yes, sir.
Okay. We are done. Keep saying. This is done. We may have discussed actually.
No, sir, just that one slide alone, sir.
Positive selection, negative selection.
Yes, this slide we have done, sir.
Okay. Yeah, then we will still, I think we will just go ahead and go faster then. Okay, it will be a kind of review maybe.
Okay, sir.
Yes. And then I have another thing also to do. So we'll go faster in this. Yes. So we have done these things. We learned about the two methods then, the direct method and the engineered stem cell method. So I hope we also discussed about gain of function. That is, you put in a gene from outside. That is, you can also call it as knock-in or transgenesis. Loss of function, you call it as knockout. You delete the gene, and that is knocking out. The other one, if you shut down the expression of a particular gene, then you call it as knockdown. And there are also temporal expression and spatial expression. There are several ways of how we put into the organism. By cell fusion, we can put transfection and electroporation. This is mostly with the cells in the culture. You can also use viral vectors in this case. That is where transfection comes in. And you can also do micro injection. This is the micro manipulator, and using a fine micro injection needle, we put in the DNA construct into the cell. And then this is how the zygote would look like. And here is the zona pellucida. This is a fine needle through which they are delivering the DNA. This is the suction pipette, which will hold on to the zona pellucida or the egg, and thus it will prevent the movement of the DNA. Movement of, or it will hold in position so that it can be pierced by this needle. Okay, and once, say, microinjection method is simple. You take the egg, you have pronuclei, that is immediately this is coming from the egg, the female, and the other one is from the male. The sperm has delivered this one here. And usually the gene of interest would be delivered into the male pronucleus, and they would fuse and form one nucleus, and then the manipulated eggs are put into a surrogate mother. We call it a pseudo-pregnant mouse. Pseudo-pregnant means we will look into it in the next slides. And once we get the pups from the manipulated embryos, we will analyze for the presence or absence of the gene. You can use PCR. You can also use southern blotting. Yes, this is the same procedure that is shown. When we are making, there are two things. First is genetic manipulation, then is cell manipulation, then we have embryo manipulation to make, right? First is we'll make the recombinant DNA, that is the DNA construct. We will put it into cells, that you can say it as cell manipulation, and then we have embryo manipulation also. So look at this. They usually would superovulate using hormones. They would superovulate a donor female, and they would collect fertilized eggs upon mating, and they would take those fertilized oocyte. You can also collect directly the eggs from this individual and allow in vitro fertilization as well. Both ways you will get fertilized oocytes, and then into it you will do microinjection. And then these embryos are transferred into pseudo-pregnant recipient female. When you say pseudo-pregnant, it would mean that they would have been vasectomized, mating between a female and the vasectomized male. So the male will perform the mating, but it will not deliver any sperm because it is vasectomized. But the female will feel, or physiologically and hormonally will feel like it's pregnant, and then whatever embryos we put on, they will be held in its uterus. Otherwise, it's not prepared for conception. And then we would get the pups. So did we discuss this part? Yes? Somebody? Pluripotent stem cells? Okay, maybe this is from a different PPT. But this is the discussion that we actually had yesterday about embryonic stem cells. A fertilized egg or a zygote would go into a morula, and then it will divide, or it will differentiate in the blastocyst, which has inner cell mass and the outer trophoectoderm. You take in the inner cell mass and you grow them in the culture plates, and then you call them as embryonic stem cells. Embryonic stem cells are pluripotent. Pluripotent means they can give rise to all the cell types that are developed from ectoderm, endoderm, and mesoderm. So that's why getting these cells is very important. But if we cannot destroy embryos, or we cannot get embryonic stem cells of an adult, already an adult, so we need different ways of how we can generate these pluripotent stem cells. For that we used, we have seen this one, right? Please tell me again. What is positive selection and negative selection we have discussed? Here is a neomycin phosphotransferase gene, and this is the gene that has been disrupted, insertionally inactivated. We have thymidine kinase here. And then when we allow homologous recombination, there are two possibilities: homologous recombination as well as non-homologous recombination. So we want homologous recombination, but we do not want the ones from non-homologous recombination. So how do we differentiate that? In homologous recombination, thymidine kinase from herpes simplex virus will not get integrated because it's outside the boundary of homologous recombination. Whereas this one, the whole thing will get incorporated, so it will get thymidine kinase as well. So both of these are resistant to neomycin because they have neomycin resistance gene in both the cases. So that is positive selection. And only one of it is sensitive to ganciclovir because of thymidine kinase, whereas the other one is resistant because it did not get the thymidine kinase gene. So that is basically the negative selection. So we we are able to separate non-recombinants from recombinants. We have three different cells: non-recombinants, recombinants with random, that is non-homologous recombinants, and homologous recombinants. When we put in neomycin, the non-recombinants are eliminated because they are not resistant to neomycin. Then we treat it with ganciclovir, and those that have got the thymidine kinase, the ones with the non-homologous recombinants, will be eliminated. And now what we have is embryonic stem cells that have incorporated through homologous recombination. I mean, gene is targeted in the particular or specific location. So now we got engineered embryonic stem cells, and we have to generate pups from it. So what they do now, we are doing embryo manipulation. We take these these are the cells that we got, the engineered embryonic stem cells, and we put them into a recipient blastocyst. The recipient blastocyst also has its own embryonic inner cell mass, right? So when you put implant and let them grow, you will end up forming a chimera, a single organism that is developed from two different embryos or cells belonging to two different embryos. And those are chimeras. And chimera is not a transgenic animal because not all parts of its body actually have the cells, have the gene, transgene. Only some of them have. So we need to generate a transgenic, first transgenic one, that is by chimera is allowed to mate with another individual, and one of it will if the engineered cells have contributed to the germplasm, they will form a gamete, and they will mate. The fertilization will happen with the other gamete here, and we might get a transgenic animal that contains the genetic modification in all nucleated cells. That is a transgenic animal. Yes, so I would now shift to where I was discussing the point that I was discussing in the last class. That is this one. So in the last class, we discussed about our goals. Our goal is to our goal is to get transgene-free induced pluripotent stem cells. Okay. How can we do that? We have identified how the OSKM factors, and they are transcriptional regulator, master regulators of pluripotency. And so we decided that we will put these genes into the cell so that they can produce large number of proteins, and efficient reprogramming would occur. But the problem is that if they are integrating, then they might be, we don't want such kind of we don't want any genetic manipulation to happen. So we decided these are the goals. We want no genetic changes should be there. We want appropriate expression of OSKM, and reprogramming should have happened. So we discussed about the design of this genetic construct. We put in the 4 genes here: OCT4, SOX2, KLF4, and c-Myc. And we also included these regions, which are self-cleaving peptides, because we want one mRNA to produce 2 different proteins or 4 different proteins. So these are self-cleaving peptides that could be put, and once a protein is produced, they would cleave themselves here, and they would produce each one part. OCT4, SOX2, KLF4, and c-Myc they would have produced. So from one mRNA, we are able to get 4 different proteins. And we also put a promoter, that is CAG, and we also have a polyadenylation signal, so that mRNA is produced with a poly A tag, and this is another promoter. And we have neomycin phosphotransferase for positive selection, and thymidine kinase for negative selection, very similar to what we have just reviewed. And it has to have its own polyadenylation signal as well. So from this part, right, from this part until this part, it is one vector transposon. We also discussed why we have ampicillin resistance gene here. So if transposase, piggyback transposase worked, it will recognize this region and the 5' end of the transposon, and it will move on to the genome and will incorporate this genome, something like this, 5', the transposon into the genome like this, 5' to 3'. The first question, why did we choose piggyback transposons? Because piggyback transposons are special. Because unlike many other transposons, they do not leave a fingerprint. What is that fingerprint? The normal cut paste transposons, or in general, transposons leave is target site duplication. These are special in the sense that if they have incorporated, they are integrated, this is a target site duplication, and so is this one. Upon excision, they cut off like this, and these two donor site, the original donor site, will come together and will form as they were originally present before the transposon was hit. Right? So these are a good choice. And as a part of our own, of this, the goal of being a genetic engineer is to follow all the basic science, all the different types of genetic elements, all the different tools that are coming up. And I mean, you don't have to know, you shouldn't, not just the tools per se. You are the one who is going to develop the tool. That means one should have been aware of all the available genetic scenarios, right? Somebody must have must have discovered these piggyback transposons. They must have worked out how it moves from one place to another place. And then somebody appreciated its beauty, that it doesn't leave its site. Then it gives a lot of flexibility for the genetic engineers to use it whenever we want, and to use it to incorporate a gene of interest. Say, for example, one second. Say this is the transposon, and we have a gene of interest here. Assume we put in OKSM here. It has incorporated, I mean, it got incorporated. So our purpose of, say, induction or reprogramming is over, and now we want to remove it. We can express transposase. So it will cut itself like this, and the donor site will form back to where they were before this transposon was inserted. And that's how we use, or we apply the knowledge from basic science and become efficient genetic engineers. Okay, and we discussed all these things, right? Yes. So we are going to go through what these people have done, and we will see a few results as well. The first thing is this is the genetic construct. Keep them close. We have taken somatic cells and put in piggyback transposon. This whole construct, we put it in there. And there are, so some of the cells will, they will get the DNA, and here we would have performed positive selection. One second. So we got some induced pluripotent stem cells because of whichever cells have gotten this, we would have grown them in neomycin. So only those cells that have gotten the DNA would have gotten this piggyback transposon would survive, and the others would not. And we still don't know which ones are in which ones we want to get rid of the transposon at a later time, right? The whole objective is this is the genome. We want to put in the transposon. We want the reprogramming to happen, and then remove the transposon. So here we are using the positive selection and negative selection slightly different from the way we used in transgenic animals. So using positive selection, we figured all these cells, or we selected all the cells that got the DNA or the piggyback transposon. And then reprogramming has occurred. They have become induced pluripotent stem cells. And we are supposed to make the genome transgene free. That means remove the piggyback transposon and not leave any footprints of this transposon. How do we do that? We can use the ganciclovir-like selection using the thymidine kinase. So that is negative selection. Those cells from which the piggyback transposon has been removed will survive in medium containing ganciclovir. And any cells, if they are still having the piggyback transposon, they will be killed, because the borders of piggyback transposon also have this thymidine kinase. Only if the whole transposon moves out, only then this transposon will, when the cells will be able to grow in this particular culture, right, in this growth conditions. So now we have transposition, we have the reprogramming complete, and we also obtained transgene free. These are all theoretical. In a research paper, we need to show proof of what we are saying. And this is from the what would have happened. And they try to show proof of this expression. They show the proof that induced pluripotency has been obtained. And then they show the proof that you can actually make these cells and make a chimera. So the first thing is to show the proof. I think in this case, this is Western blotting. Okay, I hope you know the principle. Please remember those principles properly and try to have a better look at them. And here they have, in this lane, I'm not going to explain all of it. I'll explain a little bit of each. In this lane, they have put in purified oct4. Okay. In the second case, they constructed two different ones. One is this without lin, and the other one with lin. And they want to see which one is doing better. So we have in the first Western blot here, they have in the lone, they have put in purified oct4. Second one, oct4 from the top, this one, OKSM, OSKM. And from the third one here is from the bottom one. They want to see in which case the expression is better. And so what they found is there is expression of oct4 in both the cases. Similarly, the results of SOX2 also. Also, this results of KLF and C. And if you see lin 28, it's only present in one, and it's absent here because they did not clone in this case. There is no lin here, right? Beta-actin is kind of a control to show that the cells, the amount of protein added is almost equal in all the samples, right? What would you do? We would add equal amount of protein in all the cases. Say, for example, you see little less in one case and little more in this case. What if I have added, say, 10 nanograms of protein here and 20 nanograms of protein here? Then you would find this. How would I know if it is more expression or less expression? That we can figure out if we are performing controls like this. And we should have added equal amount of protein in all the cases. Then we know that there is more expression more concentration of that protein in the given sample. I hope you understood that much. When you are looking at research papers, please look into these kinds of details very well. Those are the pillars of your research aptitude. The result is important for knowledge, but analysis of these gels, these pictures, is important for your intelligence, for critical thinking. So because these principles or critical thinking is what you will apply in troubleshooting, not necessarily the knowledge itself. I have a question and I want you to answer. See if you can figure it out. This is somewhere here, and these two are little higher, which means that they are these are little longer than this one, or larger than this one. Why do you think it is? Why are they like that? The same is the case here also. Same is the case here too, and also here. Or these two are not, they're almost same. Why is that? You see, you need to see these two are almost same, but this one is little higher. And so why do you think that is happening? Anybody? Anybody who answers correctly will get a chocolate. I'll wait for a minute. Yes? I'll give you a Dairy Milk cracker. I'll repeat the question just in case. You see that OCT4 in this one, these two are little larger than this one. Same, SOX2 also, KLF also. Maybe CMYC, these two are same almost, and this one is little larger. Lin28 is almost same. Why is that? Okay, I guess you're not trying, so I think you should be able to answer after looking at this. The chocolate bet is over, okay? Can you see anything? We are producing one large protein from here upon translation, and then they will be cleaved, right? This T2A peptide is still attached to this OCT4. It is cut off here, and the remaining part is still attached to this. SOX2 also, KLF4 also, and CMYC. And Lin28 doesn't have. Look at the gels now. OCT4, that is purified OCT4 that we took from normal cells. And these are the total protein from the samples where these two vectors were present. When they are present, they would produce OCT4 plus T2A peptide, the small one. That's why it is little larger in size. And same is the case with the other one as well here. SOX2 in both the cases also has the same. KLF4, yes, almost the same. CMYC here doesn't have a peptide. So you can almost see it to be close. Whereas in this case, CMYC has a peptide. And you can see that it is little larger. Lin28 doesn't have any, so it's almost like as if it is the purified Lin28 produced from normal sources. Okay. So that is what you should be looking at very cleanly. Now the question should also come, why is this like this? Why are they not in straight line? Those are what happens when we discuss theory and practicals differently. When we do in the lab, things will not work the ideal way. There will always be problems. The gel might be a little bit twisted. It will be tilted, and many things will happen. There is smiling like thing that will also come up many a times. That is one thing common it's observed. One more question that will get is, if we have used monoclonal antibodies against OCT4, why are these coming up? So it could be that it is non-specific. It could also be that it could be OCT4 bound to some other protein, or it's an OCT4. It could be a monomer, for example, right? Those kinds of reasoning will come if you see specific bands coming up like these things. Yes. So that's why, that is where experience matters. We need both things, theoretical and critical thinking. And in experience, because not everything works to the ideal conditions, or not, the conditions are not ideal always, there will be some differences, and we should be able to look at the gels and be able to figure out what is it. But at the same time, we should not take every band to be a non-specific band, or we should try all the possibilities and then decide what to do. We have to repeat once you performed an experiment and a gel like this has come, we have to repeat it at least thrice to be sure that it's not an artifact. And every time if it is coming, then you can be sure that it is something, it has some meaning to it, and we need to figure out what it is. Okay. So this is the same thing that we already discussed, but I want to discuss here one part. That is, we have piggybac transposon integrated, right? And we transfected transposase. So what may happen? The transposon might not have excised, may not have worked in that particular cell. So excision did not happen. Therefore, no integration will also not happen. So it is puromycin resistant and FIAU sensitive. There is another scenario where the piggybac transposon that was present on the top DNA, it might have been excised from here, but reintegrated somewhere else. So excision has happened and reintegration somewhere else also happened, because that's what transposase does. So they will be puromycin resistant and FIAU sensitive, same as these two cases. We don't want these cells. We want to get rid of transposon. So the best one is where excision would have occurred. That means transposon has come out and integration did not happen. So the DNA, the transposon might have been integrated in between before the reintegration could occur. And these are the cells that we want. If we want to show, we have to show that the cells that we got here do not have any traces of the piggybac transposon. If we didn't show, then how can we be clear or sure that there is no piggyback transposon at all? So towards this, many experiments would usually be done, and they will they do it with southern blotting. They have taken different cell lines which they have gotten after induced pluripotency, and then they have tested it with a probe from the piggybac transposon. And this show that in some cases there are traces of the piggybac transposon, whereas many of them do not have it. Okay. Okay, and that is the end. They have also performed sequencing to show that the piggybac transposon, this is the original sequence. This is where the piggybac transposon is integrated, and upon deletion, upon the excision negative selection, they have shown that the sequence has formed normal again, right? That is a proof that piggyback transposon has moved away and there is no footprint within the genome. So it is a transgene-free induced pluripotent stem cells. What I want you to do now is to read this particular paper, it is given to you in the along with the materials. Please try to take up this paper, and this is the title of it: Generation of Transgene-Free Induced Pluripotent Stem Cell Mouse Stem Cells by Piggybac Transposons. Have a look at it and read it thoroughly. Until now, I don't know how you're reading, but in the coming CAA, the questions will be like, you will ask in figure 3 of this particular paper, figure 3A of this paper, they used this kind of primer or this kind of probe, something like that. And I would ask a generation question, what does it mean? And why did they choose like that? The question could also be like, if you were in that place, what else could you have used? This means you have to think like you are the person carrying out or designing the experiments or carrying out the experiments. And I would be able to then judge your capacity to do that. So please take this paper, and there are also 3 or 4 more papers in our syllabus. I want you to read them very, very carefully. This is a way of training you to be comfortable, to become comfortable with scientific literature. And please read the method section very well, results section very well. Okay? I have covered the core of the paper, and you shouldn't have any difficulties reading it. But there are many more details, and therefore I would want you to read it multiple times. Reading one time is not useful. First time we would be able to understand maybe 30% or 40%. Then come back after finishing the whole. Whether first round, whether you understand or not, you should finish it. And then take the paper again and try to read it second time. Then you will understand about 80-90%. And the third time, you just go and have questions to the authors, like, why did you get that? Why did you use this kind of, that is where you're now raising to the excellence level, where you are finding out the thoughts of the scientist at that time and how they have solved your problem, what is the design. And that is the third time when you read it with that sense, and you will find it very comfortable. If you do like that for 3 or 4 papers, then you are good to read any scientific literature. Don't be superficial at all. When you read a paper, read it very, very, very well, right? Because you are reading for the sake of intelligence part, not the knowledge part per se. Knowledge part says, yes, you can do it like this. Oh, this is A gives B. That is knowledge part. But when you say logic part or the research aptitude is the reasoning for every step that they have done. And the scientific part would be about the biological questions to start with. What kind of, how did they get to the question? What is the question, and how did they work out for the solution? And while doing these, constructing recombinant DNA and other things, we'll have a lot of technical problems, technical issues, constraints. And there will be a lot of instances where you have to use problem-solving skills. And for that, you need to not often they would give the reasoning for it, but you can probably start imagining. When you go to the third round of reading, you probably will be in that state, and then you will be able to understand things much better. Any questions? I'll stop here. Any questions?